for joining us today for Conservation Sciences Seminar. It's my pleasure to welcome Dr. Ian Hewson. Dr. Hewson is a biological oceanographer working on the impacts of viruses on aquatic biogeochemistry. He obtained his Bachelor of Science in Botany at the University of Queensland in 1997, and then his PhD in Marine Environmental Biology from the University of Southern California in 2005. He was a postdoctoral associate at the Department of Ocean Sciences at the University of California, Santa Cruz from 2005 to 2008 before joining Cornell University as a professor. Dr. Hewson is also the director of undergraduate studies and program coordinator of for marine biology at Cornell. Dr. Hewson's love of scuba diving is what got him on track to becoming a biologist, and his interest in the great biodiversity of microorganisms and marine systems, as well as disease ecology, is what led him to his particular field of research. His work eventually led him to take on the task of understanding sea star wasting disease, which is notable for being implicated in some of the largest marine mortality events observed. I personally have followed this story as it has evolved over the years, from the rapid discovery of a single virus to a project that serves as an incredible example of citizen science and public engagement, as well as an example of the importance of the interplay between pathogens, their hosts, and rapidly changing environments. So without further ado, thank you, Dr. Hewson, for joining us, and please take it away.
Awesome. Thank you so much for the introduction, and thank you for the invitation to come and speak to you today. It's a great honor. This is my first time in Minnesota. I'm really excited. Welcome to my spare room in Ithaca, New York. I realize this is a weird format that we're in right now, but I hope today to be able to communicate some of the cool research that we've been doing on sea star wasting disease. And what I'll do is I'll just start off by sharing my screen with the obligatory thing. There we go. And all right, so today what I'm going to be talking about is sea star wasting disease, which is the tale of red herrings, misleading results, and a newly recognized disease etiology. Sea star wasting is a project that we've been involved in my lab for the last 7 years since it first started in late 2013. We have performed a number of different axes of diversity, of research, and we've sort of come to a final conclusion for our work because our funding ran out. And what I'm hoping to explain to you today is that oftentimes there are some sort of weird sort of trails that you have to take to come to a final sort of answer on a disease. And just to emphasize, this is not like the be all and end all. This is not the final result of all of our work, but it certainly provides what we think is the most cohesive hypothesis for how sea star wasting disease generates. All right, so let's see. So the key take-home messages for today's seminar are basically that apparent disease, and I put in quotation marks there because we're not clear whether it's a pathogenic disease or not, even when it's evidently transmissible, might not actually be generated by an infectious agent. It might be not pathogenic in nature. Various examples of that, for example, arthritis is not a pathogenic disease, and yet it's a disease nonetheless. Temporal changes in microbial communities and genomics can provide interesting insights into disease process. And then finally, many biological phenomena in the oceans co-vary with microscopic physicochemical changes. So looking not just at the wide scale ecosystem change that is happening at the moment, but also looking at these microscopic changes that might influence disease processes. So those are the three messages that I'd like you to, that I'm trying to communicate through this seminar. So I'll start off just with sort of a question, and I mentioned this about disease being in quotation marks. What is a disease, a condition, a syndrome, a disorder? These terms are often used very interchangeably, but many sort of imply infectious or transmissible diseases, and they often imply involvement of one or many pathogenic microorganisms. This is a statement from the the Wildlife Society on wildlife disease. And it says basically that disease is defined as any change in the normal function or structure of any part, organ, or system of a living body. It is also defined as a pathological condition occurring in a susceptible population. And then a couple sentences down, it says pathogens are natural components of ecosystems that may also be limited by environmental conditions. And so what you can see here is that it very much puts in the same frame disease and infectious agent. And that is one of the things that I'm trying to address with this seminar today. Sea star wasting is a condition which has affected about 20 different species of asteroid on the west coast of North America. We are interested in sea stars and echinoderms in general because they're both economically important, they're ecologically very significant, and they do experience these mass mortality events. All right, so as far as economic importance, echinoids, urchins are harvested as well as holothurians, sea stars less so. In fact, I'm not aware of any fishery for sea stars. However, they are consumed in certain parts of the world. They are ecologically important. So they are known as keystone species. They have this disproportionate effect upon the ecosystem structure relative to their abundance. And also other types of echinoderms are also extremely important in structuring the systems in which they live. And they do experience these mass mortality events. So if you look at history, you'll see lots and lots of reports of these boom and bust cycles of various different echinoderms. And they're known as a boom bust phyla. Echinoderms are notoriously very difficult to keep in aquarium systems, and they're very, very sensitive to environmental perturbations. And there's been a number of reports of wasting diseases in the past, including in urchins and sea cucumbers as well. But today we're going to be talking about sea stars and their wasting disease. So the big three questions that I'll be talking about is, what is sea star wasting disease? What is it exactly? How does it manifest? What does it look like? The second question, which is perhaps the $64 million question, is what causes sea star wasting disease? And then finally, I'll be addressing aspects of, well, how long has sea star wasting occurred? How long have we actually seen sea star wasting? And all three of these play into our understanding of this as a disease phenomenon. All right, so in mid-2013, I sort of had published the first ever paper on viruses in echinoderms. There was a couple of urchins that we pulled out of Hawaii that we did viral metagenomics on, and we recovered a very unusual group of viruses called the densoviruses. And I jokingly said to my colleague at Cornell, Drew Harvell, wouldn't it be great if there was a mass mortality of some echinoderm out there in the next 6 months or so? And guess what happened? Of course, be careful what you wish for, I guess. Starting in June of 2013, large numbers of sea stars started to show lesions and started to die on the Pacific coast of the Olympic National Park, which you can see here. So here you're looking at Alaska through to Baja California, and the Olympic National Park is located here just south of Vancouver Island. Pretty soon after that, we started to hear reports of the wasting disease-like condition occurring in sea stars in Vancouver, and then in Seattle, and then in the regions between, it started to spread between there. About the same time, in October 2013, we had reports from Central California, from Monterey Bay region. And then it sort of spread down the coast to Los Angeles, Southern California. It sort of missed an entire area of the coast in Oregon until about 6 months later. And then it slowly made its way up the Alaska Peninsula, Alaska, and then out into the Alaska Peninsula. So it appeared to sort of occur out of nowhere. We'd never seen anything like this in the past. And it affected a very large number of sea stars and had huge impacts upon their populations. So what does it actually look like? So we start off with asymptomatic animals or healthy animals. Here we have a photograph of Pycnopodia helianthoides, or the sunflower star. It's a very large sea star. It's very jelly-like, and it was pretty ubiquitous in the Pacific Northwest leading up until this disease event. Slowly, these animals sort of lose turgor, their internal pressure goes down, they deflate, their arms curl and twist, discoloration of ball tissues and the formation of lesions on their surface. The arms drop off and crawl away. They turn it, sort of gave it the name the zombie disease because their limbs would apparently just do it on their own. And then eventually the body wall disintegrates, the surface sloughs off, their internal organs protrude, organs and glands protrude, and then eventually the animal decomposes and turns into a pile of ossicles on the seafloor that looks like this. So it was pretty dramatic when it happened. It tended to occur in a population density dependent fashion. So you get large aggregates of these animals out there that would suddenly, one would die and then they'd all die, all right? And so it was very alarming to the public, particularly citizen science divers who were in the region. And it sort of seemed to affect them across a very wide swath of the coastline as well. And so the big question was, what causes it? And of course, this affected 20-something species of asteroid, all right? Very phylogenetically widespread disease event. These are the two most common ones at the top here. We have the sunflower star and we have the ochre star. You'll see those everywhere on the West Coast. They're still present out there today, although at reduced densities. We have Evisterius, or the mottled star, Pisaster brevis spinus, which is Patrick the starfish from SpongeBob SquarePants, And then we have various other types and species. So 20 different species were affected by this in various different intensities. Larger individuals and more active predatory species that tended to be affected first. So in other words, those which were eating other starfish. So this sunflower star goes out, it's got a very wide prey range. And also these ones, these are Solastor dawsonii, which is the morning sun star. That's also a predatory sea star that eats other types of sea stars. They were affected first and most intensively, and then the other species were affected. Like I mentioned, it's very disparate phylogeny affected. These are not closely related starfish at all right? They all fall within the Asteroidea, but they are not in any way sort of, they're very different from each other. This sort of led to, this whole disease event led to the functional extirpation of some of the sunflower star, particularly Pictopodia helianthoides at some sites in the Salish Sea. It's now being listed as an IUCN red, on the IUCN red list. And so it's considered kind of an endangered species. And it resulted in reductions from about 10 to 90% of the total population of common species, including this one, the Piasaster crassius, the ochre star. So it had this really big effect on sea star populations, affected a very wide range of them, not altogether similar or phylogenetically related. And so the big question was, well, what's causing this? What is the big factor that's leading to this mass mortality event? When we looked initially, the first thing that we did was engage with some histopathologists to understand what it looks like on a tissue level, this disease. And they took some samples of healthy and diseased starfish. They prepared these H&E thin sections of lesions. And there's no defining characteristics. So the histopathologist noted early on that there was nothing really disease defining. These are sort of normal stress responses of the sea stars. They tended to have some form of edema between their outer layer as well as the body wall. So they have this sort of spongiform skeleton, if you will, underneath their outer epidermis. So we saw some fluid build up there. There was some evidence of selenocytes with their immune cells, which are aggregating in those spaces, eventually had breakdown of the epidermis, and then you'd have infiltration by all manner of microorganisms into those broken tissues. But there really wasn't anything that was, distinct to sea star wasting that hadn't been seen before. Not only that, there were no sort of observations of any pathogenic agent that was distinctive using histopathological approaches. So for example, no cells of eukaryotic microorganisms, all right? So it was sort of no viral inclusions that would otherwise, or no sort of cell changes that might indicate a viral infection. So this was sort of a bit of a head scratcher as to what it actually was. And if you take a look at a sea star wasting lesion, this is what it looks like down a very sort of low powered microscope. This is the lesion here, down here. This is normal tissue, asymptomatic tissue of the sea star. And so when we're talking about lesions, we're simply talking about the loss of epidermis and your ability to see this underlying skeleton structure, all right? So it looks like sea star wasting somehow damages the epidermis, or it's a reflection of damaged epidermis, which then leads to sort of downstream impacts on the sea stars and their ability to fight off infiltration by other organisms. Now, if we go after this as a pathogenic disease, it should be present in the lesion margin. It shouldn't be present in the rest of the asymptomatic tissue or in clinically normal tissue away from it. And so this is kind of the sort of perspective that we took on this disease. We do know from studies of transcriptome, so this is RNA-seq studies of asymptomatic and wasting starfish, that a bunch of genes involved in structural rearrangement, autocatalytic tissue degradation, and loss of energy metabolism are upregulated in affected cells. And so this tells us that somehow this disease is basically hitting the self-destruct button, that the cells are remodeling them, or sorry, the tissues are remodeling themselves. And so this is kind of consistent with what we saw by histology. But the big question is, what is causing that? What is the environmental change, or what is the pathogenic agent that is responsible for that? And so this leads us to this big question, what causes sea star wasting? This is a topic that we've spent about 7 years investigating, many different aspects, as I mentioned, of trying to understand what's going on, including environmental change, as well as potential pathogens. We suspected early on that this was a transmissible disease. And that was based on the fact that when you take individuals from the field, you place them into captivity, they die very rapidly, especially when they become warmer, when they're subject to warmer conditions. We also noted that individuals at various different public aquaria, including Vancouver and Seattle on the West Coast, they died synchronously in their aquarium collections as the wild sea star populations occurred around in the bays around the aquarium. Very often these public aquaria, they draw in water from those bays through some sort of intake pipe. It goes through some degree of filtration. Certain aquaria do it more than others. And then it goes into their aquariums, and then it's basically put out back into the bays around the aquarium. And they noted that when divers were in the water around those intake pipes and they saw the sea stars dying, then the sea stars, and a day later, 2 days later, would start to die in their aquarium collections. And so that was sort of taken as evidence that perhaps it was a transmissible agent. Also, there was some anecdotal evidence to suggest that when you treated the in-current water with ultraviolet light, it improved the survival of the sea stars. They weren't getting as sick, or they weren't getting sick at all, compared to those that had non-ultraviolet light treated water. And the idea there is that ultraviolet light would potentially knock out any virus or any bacterium that might be coming in through those intake pipes. So we had some evidence that this was some sort of transmissible agent. In 2014, my lab performed a couple of comparative metagenomic surveys. So basically, we extracted all of the viruses from tissues of diseased and healthy specimens, and we sort of compared the types of viruses that were there. And we found this one type of virus known as the sea star-associated densovirus, present more so in the diseased starfish than it was in the healthy starfish. This was it's a very simple virus. It's basically it's very short. It's only 5,000 nucleotides long. It's not known to cause, or this group isn't known to cause disease in invertebrates at all, although they're found across a wide variety of different crustaceans and insects. And so we sort of went after this, and we were thinking, wow, that's really interesting. So we performed a challenge experiments with virus-sized material. And so we mushed up a bunch of tissue from a wasted sea star. We filtered it to get rid of what we thought were larger pathogenic organisms, things like bacteria and fungi. And we let viruses go through the filter, which is a 0.22 micron filter. We injected that into healthy starfish. And we saw that in about 80% of treated stars, it was accompanied by elevated loads of sea star-associated densovirus, and it elicited this sort of arm curling and some lesions that occurred on the surface. And so we suspected that this was a viral agent, that it was caused by some sort of virus, and sea star-associated densovirus, based on genomics, was the one that was likely to be the one that was responsible for this disease. However, so we published that in a very high-profile paper in 2014 in PNAS. In subsequent years, we were not able to replicate those initial experiments. We also started to revisit some of the data. We noted that many asymptomatic stars bore SSADV and wasted stars lacked SSADV. And so, if you think about Koch's postulates, you have to have the potential pathogen always associated with disease and not in the healthy specimens. We kind of did a little hand waving in 2014 by saying perhaps it was preclinical exposure or some other factor that was leading to it being in healthy stars. And we couldn't really explain the lack of it in the disease stars, other than perhaps it was degraded. We also failed to elicit wasting with 0.22 micron filtered homogenate in a further 8 experiments. So it was only one series of experiments, 2 different experiments in series, that we were able to do this in Pycnopodia helianthoides, the sunflower star. We couldn't do it. We couldn't repeat it 8 times later. And so we started to sort of suspect that perhaps this was not the case. Perhaps this was a spurious finding that happened to replicate in those experiments. And then we reanalyzed the data on SSADV presence in context of more metagenomics, and this sort of elucidated that there's something like 30 different densovirus which were present in that initial data set. And essentially, when we went and looked only at SSADV, it really didn't correlate at all with the disease. And so we put out this paper in 2018, which basically refuted a lot of that work that we did in 2014, and started to explore some of the environmental variables that might be driving this particular disease. It also did a little bit of work looking at bacteria, basically stating that we didn't see anything that was distinctive to wasting sea stars. If we look at other studies that were done around the same time, there was some suspicion that this might be related to temperature. Obviously, the oceans are changing and thermal expansion and heat waves are occurring more frequently. A couple of people found that higher temperatures resulted in faster wasting and death in aquarium experiments. So in other words, they would die anyway, but they would die faster under warmer conditions. Certainly observations in the field seem to line up with that. Higher temperatures accompanied wasting in the San Juan Islands, and it was sort of a threshold effect. So in other words, you had sort of a mean temperature of about 2 degrees higher than what it normally is, and suddenly they would start dying. However, having said that, there was also reports that lower temperature and lower pH accompanied wasting in coastal Oregon. You remember Oregon wasted at a different time from points further north and south, but it was a little curious as to why that was lower temperature. And so that was, one of the reasons that we thought, well, it can't be temperature explaining the entire disease phenomenon. We also started to look at variation, interannual variation in temperature, and looking at the basically the months that were preceding the onset at these different sites. And in fact, at about half of the locations, the temperature, sea surface temperature, was about, was lower than the 5-year average of temperature. So in other words, it was preceded by cooler water. It was a little bit peculiar. So we couldn't distinctively say that temperature is related to sea star wasting, but it seemed to have some interaction at certain sites. So the big question is, what causes sea star wasting disease? So armed with our knowledge that our early work, which identified sea star associated densoviruses, were probably wrong, and also looking at literature and other studies that were done suggested very inconsistent results with any environmental parameter that was being measured, temperature, for example, we didn't know where to go. And so what we started to do was design experiments that were talking about rapid shifts in environmental conditions and temperature in particular. And so in 2018, my lab deployed to the Long Marine Lab at UC Santa Cruz with the intention of looking at these rapid temperature shifts. We were going to be taking sea stars and basically putting them through a 5-degree temperature increase followed by a 5-degree temperature decrease in rapid succession, and seeing whether that elicited wasting disease, because that was consistent with some of the oceanographic buoy data that we observed at the time. So what we did is we collected 24 ochre stars from Santa Cruz. We didn't notice any wasting in the field at the time of collection. We did a pretty haphazard survey, but we didn't see anything obvious. And we put them into a common garden to try and homogenize the conditions for a few weeks. And remarkably, we went away. So we came back to Ithaca because we were involved in other research at the time. And after 14 days of being in that common garden in flow-through sea tables, you can see one here, they died. They developed wasting disease, and they all died within the space of about 4 days. All right. There were no thermal anomalies. There was no physical chemical conditions that were different from the environment. There wasn't any sort of stressors that we could identify, but they all died of this wasting disease anyway. And so this kind of set us back to square one. Again, it seems to be a common theme. All right. We didn't know, we weren't able to manipulate anything, and these things died on their own. So we redesigned some experiments. So we had some lab time out there in 2018. And so we thought to ourselves, well, what else might be going on? If it's not rapid temperature swings, there were no rapid temperature swings and these things still died, what could it possibly be? And so we started to think about desiccation. We know that this disease peaks in midsummer. Perhaps it was low tides and these things being immersed for too long in warm temperatures. So we designed an experiment where we left them out in hot temperatures for an hour and then put them back in tanks and compared them against sea stars that had not been desiccated. We looked at aquarium water replenishment rate. There's been some anecdotal evidence that if you have sort of low water quality, that actually results in faster wasting. And so faster water replenishment rates would have better water quality than lower aquarium water replenishment rates. There was some evidence maybe that perhaps it was physical abrasion. Some people had reported that they had seen pedicelaria of one species in another in the wasting lesions themselves. And so we actually took sea stars and we took pedicelaria off of one, and then we blasted them with another onto another healthy star and basically looked to see whether it generated wasting. And then we also wanted to repeat that experiment that we performed with homogenates of sea star tissue. In this case, instead of filtering through a 0.2 micron, we treated the controls with proteinase K to try and distinguish between protein-bearing material and not protein-bearing material. And the answer, looking at these results, is that everything causes wasting, of course. All right. Not one single factor could we say generated wasting and the others did not. And so if we look at this top left-hand side, we see that basically sea stars in low flow rates, they wasted faster than high flow rates. That is a statistically significant trend. If we blast them with seawater plus pedicelaria, so these are the surface features on starfish, these little claw-like structures. If you blast them with a syringe and you compare that against just blasting with seawater, they blasted, they wasted a little bit faster, although that's not statistically significant. If we dry them out for an hour, they wasted faster. And if you treat them with any sort of homogenate, including the proteinase K or the non-proteinase K, they wasted way faster than if they weren't treated at all. So it sort of led us back to square one. Again, we were trying to figure out, okay, if we can't say that it's one of these particular factors, then what is it actually being caused by? We revisited the idea that perhaps it was desiccation and temperature. Those studies which had identified temperature as being sort of a key variable in sea star wasting led us to sort of investigate whether temperature swings in the environment were as rapid as what had been seen in those experiments. And so we deployed several sensors into the intertidal zone, and temperature swings a huge amount for these sea stars. All right. So under normal conditions, these sea stars go from about 7 degrees Celsius up to 30 something degrees Celsius, all in the space of a few hours. All right. So it's really, really difficult to reconcile temperature as a sole cause of wasting, or even temperature swings, because they're experiencing this in nature under huge irradiance of the sun on a daily basis. All right. So we sort of threw that one out and said, well, perhaps that's not worth investigating. So to summarize on those sort of experiments that we ran, we knew that desiccation stress caused faster wasting, abrasion a little bit faster, proteinaceous homogenate a little bit faster, and certainly faster than no homogenate at all. Elevated water temperature based on those earlier experiments, filtered homogenates in those early viral challenge experiments that we ran. And so it seems like everything was resulting in sea star wasting, all right, which is not the answer that you want, because, being able to predict sort of things in the environment, you want to be able to say that it's a certain parameter or several, different parameters that interact to be able to generate a disease event. And so what we started to do was sort of think about, all right, well, let's look at the microorganisms that are present there, and let's just not restrict it to viruses. Let's look at bacteria, let's look at fungi, and see whether anything corresponds with this disease. A big challenge that we had in 2014 was that we were looking at carcasses of dead animals and comparing them to healthy animals. So endpoints, all right? And basically, this is really complicated, because those decaying carcasses are going to be full of all sorts of opportunistic taxa, decomposing taxa. You really can't say that anything that you find in a diseased starfish is, particularly from a bacterial standpoint, is related to the disease. You can't simply identify it based upon presence, absence, or relative abundance. And so, the other thing that we started to explore was, well, maybe this is, some sort of artificial scar or wound. And so you have to be able to distinguish taxa that you have in clinically normal tissue from sort of scar tissue, as well as that which is in the lesion. And so we performed a temporal survey of microbial communities during wasting. And so we hadn't been able to do that until that point, because every time you can't predict when wasting is going to happen. We couldn't grab samples as they started to waste in the field. And so we finally had the opportunity to do that, because we knew that sea stars would waste in our aquarium system. And so what we did is we collected animals from Davenport, just up the road from Santa Cruz. We put them into an aquarium system with the expectation that they would waste away. We subjected them to artificial scars. So we actually nicked the surface of the starfish using a sterile biopsy punch. And then we monitored for the appearance of natural lesions. So lesions that were away from those scars. And basically we sampled control tissue from an area distant from both artificial scar and lesion. And then we also sampled the margins of those artificial scars and the natural lesions. And what we were looking for are microorganisms that were present in and higher abundance, particularly within those natural lesion margins, that were either absent or very, very low abundance in artificial scars or in clinically normal tissue. And this is some wonderful work that was done by Citlali Aquino, who was an undergraduate student at UC Santa Cruz and is now a master's student at San Francisco State University. And this is what it looks like. So this is a heat map of all of the different types of viruses that we pulled out from individual viral metagenomes that corresponded with initial samples. So this is when they came out of the field. Control samples, so this is clinically normal tissue, which is taken away from lesions. And then we have artificial scar tissue, which are the margins of those artificial scars that we created on the surface of the starfish. We had 2 sea stars that didn't waste. at all throughout the course of the experiment, which was interesting. And they were fine a further week after we finished the experiment, according to people who were in the lab. And then we had 2 stars that wasted. And so the idea is we're looking for some species, some type of virus that would pop up in the natural lesions that was absent in the artificial scars and absent in controlled tissue and perhaps wasn't there to begin with. But here's the thing. We had a handful of viral contigs increase in abundance in wasting lesions over time, all right? So here you can see they were sort of relatively rare at the beginning, but they became super abundant at the end of the experiment once those natural lesions occurred. But the problem was we looked at those and they were present in the artificial lesion scars or control tissues of the non-wasting animals. And so we thought perhaps, all right, you can't really say that anything in here is maybe related to the disease simply because we find it in animals that are totally fine as well. We also noticed that many viruses became abundant late in injured epidermis. And so if you look at these sort of artificial scar margins, you'll notice that there's a lot more tiles filled in. And so that sort of suggested that something was going on with the wound repair response or some other fact that the animals are somehow resisting infection by infiltrates. That causes these viruses to become more pronounced. All right. It could also be that because the tissues are dying, viruses as a proportion of all of the transcripts that are there. These are RNA viromes, by the way. They became more pronounced. Interestingly, C-star associated densovirus was one of the contigs that we pulled out, and it didn't really have any pattern at all associated with this. So what we can say from this is that we don't have evidence for any viral contig associated consistently with wasting, but wasting may generally stimulate viral proliferation. And one of the reasons that we might have seen C-star associated densovirus associated with this wasting condition is that simply you have damaged tissue and that makes viruses more pronounced, more obvious, okay? So that sort of led us to look at bacteria. And we were curious to see what the bacteria looked like in epidermis and body walls. So we took swabs from their surface. We also did biopsy punches on a limited number of specimens because that stresses the animals as well. We noticed that those bacterial communities changed from initial to basically the time of lesion formation and the time of death, not unexpectedly. Conditions change very rapidly in an animal that's dying and selects for opportunists as well as organisms that might be consuming the decaying carcass. We looked at all the different types of bacteria and we saw a few that seemed to increase over time distinctively. So Tenacibaculum is a type of bacteroides that essentially increased between the initial time point and the time of lesion formation and the final time point. Vibrio is another one. Vibrio is often described as a pathogen. There are pathogenic species of Vibrio. However, a lot of them are simply just weeds. They're very good at taking up organic matter and they grow very rapidly when the conditions favor them. And then we found these Clostridia. And that increased from time 0 to the time of lesion formation and the final time point. This particularly, Clostridia, attracted our attention because these bacteria, Tenacibaculum and Vibrio, although they're copiotrophic, they are capable of a wide variety of metabolism as far as oxygen goes. They are both aerobes and facultative anaerobes. Clostridia, on the other hand, are obligate anaerobic. All right. And so the fact that we had anaerobic bacteria on their surface piqued our interest. That was kind of our aha moment with this disease. Why would you have anaerobic bacteria occurring almost simultaneously, if not a little bit before, in some cases, the formation of lesions? And so we sort of looked at the lesion margins again. We sort of did this in another set of experiments. And essentially what we found was that the bacterial communities, again, we had this increase in Tenacibaculum. We also noticed this increase in D-sulfovibrio, all right? D-sulfovibrio is an obligate anaerobic taxon, and Pyrrolella is a type of planktomycete, which is involved in it is a facultative anaerobe. It's actually a microaerophile, so it exists in very, very low oxygen conditions, still needs a little bit there. But we do see this consistent pattern of microbiome progression from strictly aerobic taxa to facultative anaerobes and some types of microaerophiles and strict anaerobes. And so this was kind of the thing that pointed us in the direction of what was going on. All right. So why would we find strict and facultative anaerobic bacteria on C-star surfaces? I mean, seawater is normally saturated with oxygen, particularly in the intertidal zone where there's lots of bubbles and waves crashing. And the answer is probably boundary layers. If you look at the surface of Pysastra acraceus, there's all these little structures here. These are pedicellariae, those claw-like structures. And you get these little things which are papillae, which are the gill-like structures. And so if you have enough terrain there, you can actually generate a pretty big boundary layer. And so if bacteria in that boundary layer are consuming oxygen through whatever means, then it could lead to some sort of hypoxia right on their very surface. And bacteria, what they survive on predominantly is organic matter. And in doing so, when they consume that organic matter, they deplete oxygen. And so that sort of led us to sort of study, well, wait a minute, if this is oxygen, we don't notice any big hypoxic dead zones in the area. What happens if we just decrease ambient oxygen conditions by a tiny little bit? Does that result in wasting? And so the following summer, what we did is we collected a lot of these, this species here, which is Asterias forbesii from the Atlantic. We subjected them to lower oxygen conditions. We called it hypoxia here. Hypoxia actually technically is 20% of saturated, so it's a little bit less than this. And we did that by sparging it with dinitrogen gas in very large aquarium systems. And what we found was this, all right? Sea star wasting seemed to be induced by lower oxygen conditions, all right? It didn't have to be the clinical definition of hypoxia, but decreasing it very slightly would result in this condition. And also what we did is we looked at the bacterial abundance on these in this experiment, and we found that there was a 1 to 2 order of magnitude increase in bacterial abundance over time, but in anoxic over time that was actually less than the oxic. And so there's a reason for that, and that is that anaerobes tend to have slower growth rates and lower sort of standing stock of biomass compared to aerobic taxa. And so this sort of gave us evidence, okay, so oxygen does play a role in sea star waste, or appears to. So what might be causing that depleted oxygen conditions? And can we actually measure this directly? And so we teamed up with one of the labs in the Department of Microbiology, Esther Angert, who studies Epilopycium, which is a type of bacterium that lives in fish guts, using, and her research uses these things. These are oxygen microelectrodes, and what they are is a very, very, very thin glass pipette with a cathode, platinum cathode running through them. And essentially, they're very, very expensive. They cost about $1,200 each. They're very, very fragile. You can't really bump that up against anything. And we tried to measure it on the very surface of this, which is a Pysastra acraceus. Unfortunately, what we learned very soon is that sea stars tend to move randomly. And when they do so, it's always at the least opportune moment. And in fact, we lost several of these sensors just trying to do these measurements. So case in point, don't use microsensors with animals unless you mobilize them. But what we did find is that you do have this depletion of oxygen near their surface. This is, maybe 20 microns above the surface of the Pysastra. But you find that, so this is at 0 hours, 6 hours, and 10 hours, you find this sort of decrease in oxygen leading up to their surface. And if you add glucose, which will stimulate bacterial activity, you find that increases that depletion quite substantially over time. So could asteroid surface hypoxia be a cause of wasting disease? And that was kind of the line of investigation that we pursued for the next 2 years. So the big question is, what stimulates bacterial respiration under normal conditions? Marine bacteria, they thrive on dissolved organic carbon. In coastal waters, that comes from 2 different sources. Almost all of it comes from phytoplankton exudation, primary production, excess photosynthates that get released in the form of sugars and carbohydrates and proteins and amino acids, that kind of thing. And only a very small proportion of that comes from allochthonous material that's washed in, all right? And so we started to I think perhaps this was because of chlorophyll A that was, phytoplankton. Were there, departures in the amount of primary production that might explain some of this disease occurrence? The type of, uptake of DOC generally follows very quickly from copiotrophic taxa that have a preference for lots and lots of organic matter to oligotrophs. And most of the taxa that we were seeing in our surveys belong to groups that we know are copiotrophic. They're amongst the fastest responding species to phytoplankton blooms. And so, just to illustrate how quickly oxygen can deplete, this is in the aquarium system. we had a pump failure. And basically, we add, this is with peptone, the addition of peptone, which is something that stimulates bacteria, marine bacteria. It's used in cultivation media. After only 25 minutes, oxygen had reduced almost 40% compared to a well-oxygenated system. So it doesn't take long for oxygen to go down. And bacteria that live in these coastal marine habitats do have a very quick response to organic matter input, and they are able to draw down oxygen relatively rapidly. So bacteria plankton, they rapidly assimilate and respire this organic carbon. The effect may be even higher in boundary layers on asteroid surfaces because it's relatively stagnant, and chronic loading of this dissolved organic carbon may lead to persistent hypoxia near their surfaces. So we went out to Bodega Bay Marine Lab in 2019 to see whether if we added organic carbon, would we see actually an impact upon wasting disease. We added a variety of different substrates here. You can see we had a control. We had peptone, which again is that basically additive to bacterial cultivation media. We used a culture of the green alga Denaliella tertiolecta. We also collected phytoplankton from the area by filtering large volumes of seawater onto a filter and resuspending it. And what we found is that in all of these cases, the addition of organic matter of various different forms resulted in faster wasting compared to controls. So this kind of gave us some evidence that, it is related to organic carbon. Incidentally, we also saw some big shifts in the abundance of bacteria that seemed like they went through this very strong increase in abundance. And then pretty soon after that, they crashed out in abundance. We believe that that probably relates to oxygen. As I mentioned, aerobic bacteria tend to become really, they grow really quickly. And then as anaerobic conditions start to set in, their growth efficiency goes down. And so you find this sort of decrease and increase in anaerobes, which are growing much more slowly. So this sort of suggested that DOC enrichment can actually cause wasting. So the question is, why are sea stars so susceptible to hypoxia? Unlike most marine organisms of their size, these organisms rely on passive respiration. They don't ventilate. They don't have lungs. They don't have gills that they pump water over. They actually rely upon diffusion of gases coming from seawater into their bodies through their boral surface, their upper surface, and through their tube feet. Sometimes they can wave their tube feet a little bit more, but if they're attached to rocks, that doesn't really make a big difference. And so what you tend to find is that larger individuals have a very much higher respiration rate per volume. And larger stars basically have a bigger oxygen demand per unit volume than smaller stars. We also, can calculate their relative, their absolute rates relative to what you can hypothetically diffuse across their surface. And what we found is that those species that died during this wasting event, they don't have a lot of leeway as far as their ability to take up oxygen. They are basically having to respire at their hypothetical maximum, whereas those species that were less affected by this wasting disease, they have a lot of leeway. They're only respiring a fraction of what they can possibly, breathe in through their skin. And so the other really interesting thing that we came across is that more rugose stars are more susceptible to wasting. And what we started to notice is that those species that are flat, that are relatively smooth on their surface, they didn't really have much wasting at all. Some of the species were unaffected by wasting. Whereas those species that were really complicated surfaces, they were the ones that were dying from this. So for example, Dermasterius, the leather star, a very smooth starfish on the surface, had almost no wasting disease. Whereas co-occurring in exactly the same sites and same environments, these two species, Pycnopodia and Paisaster, lots of wasting, and they're almost gone now, all right? Obviously, the longer you have surface processes, the more rugosity that you have on their surface, the more extensive a boundary layer you're gonna have. So this is a figure that I pulled from a paper on seagrass. What you tend to find is that if you have short seagrass blades, you tend to have sort of a less extensive boundary layer. Whereas if you have much longer blades, you have a more extensive boundary layer. And so we thought perhaps this might relate to interspecies susceptibility. How do you compare rugosity? Well, we went to a computed tomograph scanner, a CT scanner, and we measured the rugosity properties across all of the affected species. And that was done computationally by building big models, 3D models. And what we found is that species that were more affected by wasting had a very much more rugose surface than those which are not affected. And the idea here is that if you have more of an extensive boundary layer, there's more potential and a longer distance over which oxygen has to diffuse. And if there's depleted oxygen within that surface layer, it's going to basically block respiration of the animals. So they effectively suffocate. So big question is, do we see evidence of this effect in 2013? This is all very well. We're doing this in 2018 to 2019. Was it at play in 2013? And we do actually have some evidence that this was the case in 2013. If we look at, for example, the types of bacterial signatures that we had in RNA-seq libraries, what we tended to find was that strict aerobes tended to sort of not be present in disease specimens, whereas in disease specimens, we actually had some strict anaerobes that suddenly appeared. Likewise, some unpublished work that we did with quantitative PCR and bacterial taxa, we found that over time, these groups of microaerophils and facultative anaerobes, and copiotrophs for that matter, they increased over time. And so we do see some very consistent patterns with what we had in 2013. There's even more evidence of hypoxia, all right? Larger stars died faster than smaller stars. The surface area to volume of sea stars, which dictates their ability to take up gases from the environment, is very much smaller for larger animals, all right? And so that sort of is consistent with this idea that oxygen's playing a key role. In lesion time in experiments, it's most strongly explained by animal volume and aquarium flow rate, all right? More active and predatory species, which obviously consume more oxygen because they're moving around a lot more, they died faster than less active species. And then when we started to look at some of the transcripts themselves from the host, what we found is that high affinity cytochromes and, high oxygen affinity cytochromes, particularly this one P452J6, was highly enriched in wasted star compared to asymptomatic transcriptomes in at least 2 different studies, right? So this is some genetic evidence that in fact, this disease is actually a result of oxygen starvation. Even more than that, we started to explore, so obviously we can't go back in time to 2013 and 2014. We don't have time travel yet. We're still working on it, all right? It's never gonna happen. But what we did have, and unfortunately we lost most of our specimens that are capable of being used for molecular work through freezer failures. I'm sure everybody's experienced that in the past. We had a really bad one that went on for 2 weeks and we lost all of our sea star carcasses, or at least made them unamenable for RNA and DNA work. But what we were able to do is think about signals in tissues that were not affected by freezing and thawing that might actually give us an indication of processes that were happening. And so we turned to this, which is stable isotopic composition. And the idea here is that under most conditions, there's a balance of 15N to 14N in nitrate and other inorganic nutrients. Microorganisms under anaerobic condition enrich the 15N signature that's basically in tissues or is free in the environment. And so did we see a signature of these anaerobic conditions in 15N in those affected and unaffected tissues at the time? And the answer is, if you squint, yes, we did. We didn't find a significant result, but they were generally higher, okay? I'm not gonna profess that that's the final result of that because it's not statistically significant, but we tended to find higher signatures of 15N in these affected stars compared to these unaffected stars. When we look at sort of variation of sea star wasting over time, we looked at some physicochemical conditions. It turns out that nobody was actually performing long-term monitoring of sea stars and sea star wasting in concert with environmental variables. These are really difficult to measure consistently, and buoys generally don't have the ability to have these sensors on them. But what we did find at one site where we had a citizen scientist in the water every single week monitoring sea stars in the same length of dive was that the peak intensity of wasting tended to happen right after the peak in chlorophyll A concentration at this one site, as well as the minimum of dissolved oxygen, all right? And here looking at the time of mass mortality, chlorophyll A was way higher than compared to other months, and oxygen was less than other months. So there's some evidence from the field that this effect might be occurring. Other sort of things that might have happened in 2013. We looked at precipitation data in concert with the time of wasting onset, and what we found is that in several of the cases, we had a drought condition that was occurring on the West Coast. There was very little precipitation. And then when wasting actually occurred, that precipitation kicked off, all right? One consequence of that is that when you have a big rainfall after a drought event, it brings into the coastal zone a lot of organic matter, and all of that tends to be very highly labile to marine bacteria. So it looks like there were some weird conditions that happened in 2013 that might have corresponded with this disease. Also, if you look at satellite imagery, so this is the CWIFS imagery, this is Aquamotus imagery, looking at chlorophyll A concentration from 2010 through to 2016 in 3 different months that we knew corresponded with wasting. you tended to sort of see, if you squint again, you can see that there's increases in chlorophyll A at those affected sites in those years compared to other years, more so than others, particularly here in Central California. All right, in September, there was an onset. There was the highest upwelling ever noted at that location at the time. If we look at absolute upwelling indices, upwelling brings inorganic nutrients to the surface and fuels phytoplankton blooms around coastal areas. Again, we tended to find, like in months preceding the wasting onset, there was higher upwelling. This is even more pronounced when you look at this as a total amount of upwelled water in the preceding 6 months. So there seems to be some correspondence between environmental conditions and wasting onset. And I'll just skip over that. Just to mention also that under anaerobic conditions, there's other effects too. Ammonia, which is normally detoxified by ammonium oxidizers, tends to be inhibited under anaerobic conditions. And so sort of this runaway effect of toxin accumulation of ammonium toxicity. And so what we tended to find with this is that under disease, within diseased starfish, we had a very much lower abundance of organisms with ammonium monoxygenase. And so it's likely that there was some impact of toxins as well. And so this is kind of a conceptualization of the disease as we understand it now. Essentially, under normal conditions, you have dissolved organic matter, which is being exuded by the starfish in the form of mucus that stimulates bacteria. Normally, that doesn't result in any inhibition of oxygen making it into the animal itself or carbon dioxide being released. Also, you have this exudation of ammonia by the starfish, which is detoxified by ammonium oxidizers. But when you have extraneous amounts of dissolved organic matter, it fuels bacterial activity. It makes that boundary layer more extensive. Oxygen has a harder time getting to the animals. They suffer from an oxygen deficit. Carbon dioxide accumulates and increases that hypoxic condition. And also ammonium tends to increase within the exterior, within this boundary layer. So lots of negative impacts of this potential model on sea star wasting. So I just want to end up this talk just with a minute here talking about how long sea star wasting. I mentioned that sea star wasting first appeared mysteriously in 2013. But the truth is that it's been around for a really, really long time, all right? So this is from a paper in 1898. And what you'll see here is a great description of sea star wasting as it occurs in today's ocean, all right? The disease attacks both the common star and the purple star. It attacks the skin first and not infrequently eating its way through the body wall, all right? So it looks as though this has been around for a while. When we started to look at occurrences of sea star wasting, perhaps by different names in the past, we started to see more and more reports. Here's a specimen of astropecten from around Denmark. This is a sea star wasting. It's got an erosion on their surface and their body wall are proceeding. This is from the Mediterranean Ocean. Again, you see these sort of lesions forming on their surface. And this was from the late aughts. When we looked around the world, the Yellow Sea in China, in Australia as well, Antarctica, New Zealand, when I was there on sabbatical, you tend to find these sort of wasting-like conditions all over the place. It's just they're sort of not that pronounced, but it seems to be a normal sort of condition that happens to have been exacerbated in 2013 and 2014. So here's a total timeline of it. 1898, that should be the first report. And then leading up into sort of from the 1970s until the present day, a huge number of sea star wasting-like diseases occurring. So it seems to be a relatively common phenomenon. Okay, and so just to summarize for this talk, so sea star wasting, a disease, a condition, a disorder, or a syndrome, it's likely a manifestation of boundary layer hypoxia. That's our hypothesis at the moment, which is driven by locally enhanced bacterial activity that drives down oxygen concentrations. The source of the organic matter is probably phytoplankton. It could also be decaying carcasses of nearby starfish, which might lead to some sort of density-dependent observation in the field. The effect will rapidly snowball when populations are dense because of lability of decomposition products. And the effect will be worse under climate change because higher temperatures lead to faster bacterial activity, bigger drawdowns of oxygen. So sea star wasting, we believe, is a disease, but is not likely pathogenic or transmissible. All right, so that's a key take-home point. It looked like a transmissible disease caused by a pathogen. We now do not believe that to be the case. We believe this to be an exacerbated natural condition from environmental variation. that happens to have been really intense in 2013. And with that, I'd like to thank, I apologize for going over time just by a minute or two, but just like to thank my graduate students, undergraduate researchers, and fabulous people who've been working in my lab for a while, as well as my funding sources through the National Science Foundation. I'd be happy to answer any questions that have come up.
Thanks so much. So I don't see questions right away in the chat box. So just a reminder, if you have questions, go ahead and drop them into the chat box and I can read those out loud. But maybe to get things started, I had a question. I was wondering if, are there like any sort of like treatments for like aquarium populations or like solutions for wild populations that are affected by sea star wasting disease?
So I can answer the first one really easily, and that's been successful. So two of the ways by which aquariums have been treating sea stars that show this disease are to treat them with antibiotics, which appears to work for a short amount of time. And then as soon as you withdraw the antibiotics, it comes back. And with the antibiotics, that is obviously knocking out bacteria of certain types, depending on which antibiotic you're using. So one of the things we've been encouraging aquariums to do is essentially oxygenate them a lot, right? So turn up your bubblers, turn up the amount of dissolved oxygen that's in there, stir it a lot, create water motion, break down those boundary layers on the surfaces of sea stars. And that has actually been successful anecdotally at 4 or 5 aquariums. They've managed to keep it at bay. Getting rid of dissolved organic matter within their aquarium system is either through water changes or one of the things you can do is treat it with ultraviolet light. You remember I talked about like UV light seemed to sort of get rid of this condition. UV light does tend to sort of break down organic matter and make it less bioavailable. And so that might be one strategy that people can use. As far as the field goes, it's a little bit more complicated. I think it comes down to changing ocean conditions, climate change, and just sort of preventing blooms, doing our best to sort of curb emissions and stop increased stratification and temperature rise. But yeah, not sure if that answers your question.
Yep. Oh, it does. But actually, one little tag onto that is that you mentioned that allochthonous input is probably is a lot less than the autochthonous input of DOC. But I guess are areas that are affected by sea star wasting disease, are they associated with eutrophic coastal waters, or was that just like a kind of one-time thing in those satellite images?
As far as sort of coastal eutrophication, anthropogenic eutrophication, there's a couple of anecdotal reports from near Seattle where it tended to be more intense in sort of waters that were affected by stormwater runoff and sewage outputs. But realistically, what we're looking at is less of a sort of anthropogenic eutrophication, just more kind of natural cycles of stratification, upwelling, bringing in organic nutrients, and then that is what's causing elevated primary production. These are not sort of algal blooms like pea soup green blooms that you see, or red tides or harmful algal blooms. This is just elevated productivity that is sort of delivering more organic carbon to it. So as far as like human impacts on this, there certainly at very localized sites, there might be some impact of human-derived organic materials, but it's not most of it.
Great. All right, well, it's about time for us to wrap up. So thank you so much for that great presentation, Ian. And see you all next week.
Thank you so much.
Yep.
Right.